Good day everyone, I am Pauline Langido and today I will impart about the cell motility, the integrating role of the plasma membrane. Today I will be going to tackle about the membrane curvature and membrane transport and flow. What is membrane curvature? So it refers to the physical bending of membranes to accommodate various cell morphology changes as well as the formation of membrane-bound transport intermediates like spherical vesicles or tubules. As membranes are inelastic and resistant to spontaneous bending, membrane curvature is generated through active processes mediated by specialized proteins, lipids, and the cytoskeleton. A motile cell contains regions characterized by high membrane curvature, most notably the leading edge of the cell, which has a radius of curvature of 50 to 100 nanometer, and intermediates in membrane transport including intracellular vesicles and endosomes. The motility process involves continuous reorganization of the morphology and curvature of these membrane domains. The spontaneous curvature of a membrane and its bending elasticity are determined by the composition of the membrane. The local curvature of the membrane is determined by this characteristic together with the forces acting on the membrane including cytoskeletal generated forces and pressure gradients across the membrane. Rather than being a passive consequence, local membrane curvature is dynamically determined by a continuous interplay between shape and composition. The curvature-sensing lipids and proteins respond to membrane curvature and at the same time induce spontaneous curvature by energetically favoring curve morphology and by inducing changes in local cytoskeletal dynamics and force generation. The structure of lipids and membrane proteins and their interactions can favor membrane deformations with shape lipids or protein incorporated within the bilayer induce local curvature. The correlation between lipids composition and local curvature is clearly demonstrated by in vitro experiments in which giant unilamellar vesicles composed of several lipid species spontaneously phase separate into domains which vary in both composition and local curvature. Furthermore, expression of membrane proteins, for example the Shiga toxin in cell, can induce the formation of highly curved membrane tubules in which the toxins is highly enriched. So the protein with domains that insert into the bilayer, for example an amphipathic alpha helix domain, can also sense and induce curvature. Scaffolding proteins that bind to the membrane provide yet another important mechanism for inducing curvature. One of the best characterized families of scaffolding proteins is the BAR domain protein family. These proteins contains a rigid curved domain which interacts with membranes via hydrophobic and electrostatic interaction. Different BAR domain proteins are characterized by different natural curvature. In particular, FBR and NBR domain proteins induce positive curvature, whereas the IBR domain protein induces negative curvature. So in general, the action of curvature favoring lipids and proteins and scaffolding proteins is viewed as being highly cooperative. Membrane deformation in cells typically involve a very large number of curvature, sensing and curvature-inducing molecules. So the leading edge of motile cells is characterized by a thin protruding region with high membrane curvature. This thin structure is maintained by restricting actin polymerization to the apex, and it is clear that localized pushing forces induced by growing actin filaments are important for generating and maintaining this high curvature. Membrane transport and flow. So the transport between internal membranes and the plasma membrane through endocytosis and exocytosis are essentially the only way for lipids and transmembrane proteins to enter or leave the plasma membrane. Endocytosis includes several different processes including clathrin-mediated and cavillar endocytosis, by which patches of the plasma membrane invaginate inward and bud off to form vesicles, thus reducing the area of the plasma membrane. The membrane transport between internal membranes and the plasma membrane accompanies movement in all cell types. The overall rates of transport between internal membranes and the plasma membrane vary among different cell types. In Dictyostelium amoeba, it takes 3 to 10 minutes to replace an area equal to the cell surface area, whereas in fibroblasts, it takes approximately 1 hour. So the rates of endocytosis will depend on the availability of internal membranes, which also depends on membrane biogenesis. The inhibition of membrane trafficking in several cell types, for example, the fibroblasts, endothelial cells, and dictyostelium, reduces persistence in migration and reduces cell speed. One of the main functions suggested for membrane trafficking is the recycling of membrane components, such as cell adhesion molecules. Other possible functions include lipids transport to induce membrane flow to the leading edge and the establishment and maintenance of cell polarity. The importance of membrane transport to motility probably varies among different cell types, and research is needed to clarify its functions. So the cell movement requires continuous recycling of essential membrane components. For example, the membrane proteins such as integrin, which couple the actin cytoskeleton to the substrate, must be continuously replenished at the bleeding edge as the actin flows rearward in the cell frame of reference. So a continuous flux of bound integrins moves with it. To maintain motion, there must be a counterflux of adhesion molecules entering the leading edge. So the recycling of transmembrane proteins, for example, the integrins can occur via several mechanisms. So within the bilayer, the passive diffusion can contribute, but it is probably too slow to compensate for the retrograde flow of adhesion away from the leading edge. Thus, at least in rapidly moving cells, active transport is likely to be needed. Directed motion through the membrane can be driven by bulk membrane flow or motor-driven transport. The membrane transport via polar membrane trafficking, in which integrin containing vesicles are targeted preferentially to the leading edge, can provide for directed transport. So, in most motile cells in which membrane dynamics have been directly measured either by single particle tracking or lipid-bound particles or by photobleaching or photoactivation, no net membrane flow has been observed. In this figure of membrane transport and recycling of adhesions, in letter A, where adhesion complexes assemble in a hierarchical fashion from integrins embedded in the membrane and cytosolic proteins, nascent adhesions form at the leading edge and attach to the substrate. Actin network growth and flow away from the leading edge leads to a rearward flux of adhesions. In letter B, where various possible mechanisms for recycling integrins back to the leading edge have been suggested. Passive transport by diffusion will tend to equilibrate the integrin density but may not be fast enough for efficient recycling. Active transport to the leading edge can occur with the help of motor proteins which drag the integrins within the membrane, by drift induced by bulk membrane flow, or by polarized membrane trafficking. Changes in the area of the plasma membrane accompany movement in some cell types. Because the plasma membrane is essentially inextensible, such changes must be generated by an imbalance between the processes of exocytosis and endocytosis. During motility of Dictyostelium amoeba, substantial dynamic changes in surface area have been observed for individual cells over time, whereas rapidly moving fish keratocytes have essentially constant surface area as they move despite changes in their overall morphology and speed. The function of surface area changes during motility is still unclear, whereas in Dictyostelium amoeba, it seems that surface area regulations is an active part of the motility process. In keratocytes, membrane area remains fixed and the membrane seems to be stretched thoughts around the cytoskeleton, imposing constraint on shape and movement. So the rapid changes in cell surface area can also be mediated by budded, unfolded membrane domains that acts as a membrane reservoir and enable cells to respond to sudden changes in membrane tension. To prove the membrane behavior in the motile fibroblast, membrane tethers were extracted from the cell membrane. The initial force required to pull tethers was found to be independent of tether length, indicating that the membrane flowing into the tether was taken from a reservoir at same point. The tether force started increasing abruptly in an exponential fashion, indicating depletion of the reservoir. Overall, the question of how cells regulate their surface area and how changes in surface area relate to the motility process are still largely unanswered. Although a cell probably cannot directly measure its surface area, it is thought that cells respond to changes in membrane tension. Thank you for listening and stay safe, everyone.